Welcome to video 2, Mode of Action of Enzymes. How does enzyme work? So what you see here is an enzyme, and every enzyme has an active site, which is a depression in the enzyme. The substrate will bind into the active site of the enzyme. As you see here in the clip, the substrate will bind into the active site of the enzyme to form the products. Another thing to notice is that when the substrate binds to the active site of the enzyme, an enzyme-substrate complex is formed. The products will then leave the active site of the enzyme. So let's look at step by step how does enzyme work. So the substrate will fit into the active site of the enzyme perfectly. This will form a complementary fit. A complementary fit is essentially a perfect fit between the substrate and the active site of the enzyme. This will form the enzyme-substrate complex. When this happens, enzyme will be able to speed up the breakdown of the substrate. The products will then leave the active site of the enzyme, and the enzyme is free to receive the next substrate. Now, how does enzyme catalase work? We've already seen some examples of sources of catalase, that is from potato or liver cells. Catalase will break down hydrogen peroxide into water and oxygen. So, using the mode of action of enzyme, we are going to look at how enzyme catalase work. So, enzyme catalase has an active site shape that is specific to the substrate hydrogen peroxide. Hydrogen peroxide will feed into the active site of the enzyme to form an enzyme-substrate complex. Catalase speeds up breakdown of hydrogen peroxide, and the products, which are water and oxygen, will leave the active site of the enzyme. Catalase is now free to receive another molecule of hydrogen peroxide. Enzymes are specific in their action. So for example, enzyme A will have a shape of active site that is complementary or specific to the shape of substrate A. And enzyme B, for example, will have an active site with a shape that is complementary to the shape of substrate B. And enzyme C will have an active site with the shape that is complementary to substrate C. So each type of enzyme will act on a specific type of substrate. Shape of active site of enzyme is what we say complementary to the shape of substrate. Let's now check for your understanding. So you already know that yeast containing enzyme catalase, when added to hydrogen peroxide, will result in bubbling. But what if we now add yeast containing enzyme catalase into oil instead? What would you observe if yeast is added to oil? Those of you who predicted that no bubbles will be produced, you are right. But why is this so? Why oil cannot be broken down by enzyme catalase into oxygen and water? So as you can see here, enzyme catalase has a specific shape of the active site. An oil molecule has a shape that is different from the shape of the active site of the catalase. So this means that the oil molecule is not able to bind into the active site of the enzyme to form enzyme substrate complex, which means there's no complementary fit, therefore no reaction. So let's put it in words. Why oil cannot be broken down by enzyme catalase into oxygen and water? No complementary fit between the oil molecule and the active site of enzyme catalase. Hence, no reaction takes place.